Hello students, today we are going to start with phylum Porifera, its characters, classification and canal system in Syscon. So these are pore-bearing animals. Pores means holes, okay, and ferine means bearing. So this is the bifurcation of the terminology and are commonly called as sponges. As we all know, they are lower metazoans placed in subphylum Parazoa. So the name of the subphylum is subkingdom is Parazoa and study of this is called parazoology. In majority, these are marine. Exceptions, one or two cases. Some are freshwater also. Total, there are 150 species discovered till now, and their fossils has also been reported from the rocks which are found in marine habitat underwater, and they are known to be 600 million years old. These are plant-like organism, but their animal nature was first discovered by Ellis. So the name of the scientist, the discoverer is Ellis. And the term Porifera was coined by Robert Grant. So remember this, it can be asked during the exam. Now let's move to the journal characters. Body forms. Body forms may be like vase-like, जैसे flower pot होता है ना. And the example is leukocytes, Scypha. They may be cylindrical like Euplectella, spherical, Hyalonema, Euspongia, or irregular mass of branches like Spongilla. Uh, you have uh, you have studied in class 12 that there is Spongilla, okay, and this is asymmetrical. Now, organization, they have cellular level of organization, that is, they are multicellular but showing no connect contain coordination. Cell aggregate plan is little differentiated, so there is not very much differentiation. Size, they may vary in size. Size range is 1 centimeter to 1 meter, so they can be 1 centimeter also, and they can be as long as 1 meter also. Now let's move to the symmetry. Sponges are radially symmetrical. That means they can be cut from any plane into equal halves. Example of these are Scypha, Euplectella, and they can also be asymmetrical. Example, Spongilla, I have told earlier. Now, the body forms, they are diploblastic, developed from two germ layer. That is, any of the two. There are three mainly germ layer, that is ecto, endo, and meso, but only two contribute, so they are diploblastic. Body wall, adult sponge also formed of two layers. Outer is dermal layer or is called pinacoderm, and it is made up of pinacocytes. And the inner gastral layer, this is called choanoderm, which has flagellated collar cells and which exhibit water current. Choanocytes are present only in sponges. Between the two layers, there are many amoebocytes as well. Okay, so there are different types of amoebocytes depending upon the kind of function they exhibit. First is chromocytes. Chromo means color, so they are known to be pigment cells. Now, trophocytes. Tropho means trophic. That means they contribute to the nutrition. Now, thesocytes. Theso means food storing cells. Collencytes, these secrete collagen fiber. Myocytes. Myo means muscles. Okay, so they are contractile cells. Muscles का काम क्या होता है? Contraction and relaxation. So they are contractile cells. And they form sphincter around the dermal ostia. Dermal means the dermal skin layer, and ostia, the entry, oscula, exit, phagocytes engulfing the excreta and damaged cell. Phagocytosis. So phago means the what cells? Engulfing cells. Okay. Sclero means spicule forming cells. Spicule are ये thorn-like structure. Archaeocytes, totipotent cells. Okay, what are totipotent cells? They're the cells which have capacity to regenerate and reproduce. Now, let's move to the pores. The body wall is porous, means it has holes. So, as we all know that their organization is not very, you know, the skin tightening is not very much firm. So, there are pores. There are two type of pores, numerous and small size. So, they are called dermal ostia or inhalent pores, and single or few large size pores are called osculum, exhalent pores. So, the dermal ostia form mouth, and the osculum forms anus. Okay, they act as inlets and outlets of water current. Inlet one is dermal ostia and outlet one is osculum. So they're analogous to mouth and anus, as told earlier, in a higher organism. In these are lower organism. In higher organism, they form mouth and anus. Mouth for entry of the food and water, and anus for the exit one. Now Let's move to the habitat. Majority of sponges are marine, except single family of about 150 species of Spongilla is well known as freshwater sponge. Some species, majority are marine, okay? That is, they live in saline water. Saline water, they exhibit osmosis as well through the skin surfaces. Okay, and in freshwater, Spongilla is a freshwater, and it's an exception. So remember that. Now, as you can see the diagram, this diagram is a body wall of sponges. So in exam, you must know how to draw the diagram. These are amoebocytes. These, the border of the pores. Now, tri-radiate spicules. As I've told earlier, spicules are thorn-like structure. Tri means three. Radiate means radiating. So तीन direction में radiate करता है. That's why it is called tri-radiating spicule. Porocytes. These are porocytes. Ostrium, the entry one. Okay, and now this is a scleroblast. What are scleroblasts? I've told earlier. Scleroblasts. You can comment what are scleroblasts. I've told earlier. Now this is mesoglia, or also called mesenchyme or mesophyll. This is situated between the dermal layer and pinacoderm of the flat pinacocytes. Okay, and the another layer is inner gastral layer. I told earlier, coenoderm of the flagella collar cells. Flagella beat constantly but uncoordinately to maintain the water current. They move constantly but uncoordinately. Uncoordinately इसलिए क्योंकि जिस डायरेक्शन में वाटर करंट जाएगा मेजोरिटी वहां को ही मूव करेंगे. A few exception cases are also there. Now, the next thing is gelatinous and non-cellular. So mesoglia is gelatin, gel-like layer, and it is non-cellular. Okay, it is gel-like, so it is non-cellular, and it has many amoebocytes. I have discussed earlier the types of amoebocytes. Now, let's move to the coenocyte. So this is the diagram of coenocyte. This is the flagellum. Okay. Next one is collar, blenophlase, rhizoplast. This is parabasal body. Parabasal means because it is situating at the bottom. This is the nucleus, membrane, cytoplasm. Now, next move to the next diagram, that is of gamete. This is an important diagram of gamete because it has archaeocytes. Okay, archaeocytes. This is the outer membrane, micropyle. In plants also there is a micropyle, and in this animals also there is this micropyle. So you can remember that the entry point, okay, of the mouth is called micropyle, and monoxon spicules. Mono means single, exon spicules मतलब सिंगल थॉर्न लाइक. जैसे इसमें ट्राई रेडिएट था ना, मतलब तीन तरह से रेडिएट करे थे, बट इसमें सिंगल तरह से रेडिएट करे, एक ही डायरेक्शन में रेडिएट करे. ओके, and next is inner membrane. So remember this, this is the inner membrane and this is the outer membrane. It's a very simple thing. Now let's move to the other characters, that is organelles of locomotion, but there are no organelles of locomotion, and they are sedentary. Sedentary meaning they cannot move from one place to another. Okay, so they do not have any locomotory structures. Now the canal system. This is a very important topic. Canal system in sponges. All sponges have a peculiar system of interconnected canals called canal system. Water current is moved, so it is also called aquiferous system. क्योंकि इसमें water current, aqua means water ना, ferrous means ferricing. So you can bifurcate the terminologies and understand the thing. The central cavity of canal system is called spongiosole or paragastric cavity. Canal system may be asconoid type, characterized by unfolded body wall, large spongiosole lined by coenocytes. Example are leucosolenia and clathrina. Now, the next is cyconoid type, characterized by presence of incurrent and radial canals formed by the folding of body wall, collar line cell only in the radial canals. Example, type and grantia. The next is leuconoid type. The third type is leuconoid type, characterized by presence of flagellated chambers lined by collar cells. Example, euspongia and spongilla. So there are three types of canal system. You must remember asconoid type, cyconoid, leuconoid type. Next is endoskeleton. So what kind of things endoskeleton is made up of? Most sponges possess an endoskeleton secreted by amoebocytes. It is formed of either calcareous spicules. Calcareous means calcium, formed of calcium carbonate and secreted by calcoblasts. The cells which secrete this calcium carbonate are called calcoblasts. Example, Skypha. Siliceous spicules. Siliceous means having silic acid and secreted by silicoblast. Blast means cells. Example is Euplexella. Next is spongin fibers. Threads form of spongin protein and secreted by spongioblast. Example, euspongia. The terminologies are very much interrelated, so you can remember that with the one or the other thing, or combination of siliceous spicules and spongin fiber. So there is also a combination of silica and spongin fiber. Example, Chelina spongilla. Endoskeleton forms the basis of classification of sponges. So endoskeleton is also very much important. These two topics are very much important. The first one is the canal system and second is the endoskeleton system. These are asked in exams, in midterms as well. Okay, so remember that. Now let's see the nutrition. Sponges are omnivorous and holotrophic. What do you mean by omnivorous? That they feed on both plants and animals. And holotrophic means they can also prepare their own food, own nutrition. These are chiefly feed on planktons. Now, the main food choice is plankton. क्यों? क्योंकि plankton ज़्यादा easily available है उनके surrounding में. क्योंकि वो move नहीं कर सकते ना, तो वो तो वही चीज देखेंगे ना जो उनको आसानी से मिल जाए. तो प्लैंकटन उनको वाटर करंट्स में आसानी से मिल जाता है, तो वो वाटर करंट्स में प्लैंकटन को अब्जॉर्ब करते हैं अपने डाइजेस्टिव सिस्टम में. न्यूट्रिशन कम्स respiratory water current is maintained by constant beating of the flagella. Okay, or collar cells. These are called ciliary or filler feeders and show intercellular digestion. What kind of digestion they show? Intercellular, that is among the cells. Reserve foods material is stored as glycogen, fats, glycoprotein, or lipoproteins. That is lipids and protein. Special amoebocytes called theocytes are also present. Now let's move to the respiration and excretion. So the respiration is aerobic and excretion is ammonotelic, that is they release ammonia. Respiration is occurs through general body surface and excretion also occurs through general body surface. Now circulation, food is distributed by cell to cell diffusion and by special amoebocytes called trophocytes which move in about mesoglia. Now, next is sense organs and nervous system. Sponges do not have any special sensory or nerve cells. So there is no coordination of action of the whole body, as I've told earlier, because their cells are not much differentiated, but the individual cells are sensitive and react individually. They show slow and local restriction which are sufficient to fulfill the needs of sedentary sponges. Here it is again and again mentioned that sedentary because they cannot move. Okay, so this is a plant characteristic. Okay, but they show circulation, respiration, excretion, and digestion as well. So this is a animal-like character. So they exhibit both the characters. You must see it carefully. This sensitivity is highly developed and some kind of coordination has been reported among the myocytes surrounding dermal ostia and ocula. Now let's move to the reproduction and development. So both asexual and sexual reproduction. In sexual reproduction occurs generally by external budding. In example, types and leucosolemia. Formed by internal buds called gametes. Example, spongilla. Each gamete is formed of arcusites. एक gamete बहुत सारे arcusites से बना होता है. ठीक है? Surrounding by a protective covering of amphidiscule. Amphidisc का क्या मतलब हुआ? Amphi means a disc-like structure. In amphibians, there is a disc-like, so it is related to that thing. And they have a slit-like aperture called micropyle. Slit-like मतलब कि पतला सा जो aperture होता है. Slit मतलब कि slit क्या होता है? एक पतला सा एक linear छेद टाइप होगा. ठीक है, उसको माइक्रोपाइल बोला जाता है. हमने देखा ना पीछे एक माइक्रोपाइल कैसा होता है, वो उसको बोलते हैं स्लिट लाइक aperture. Most of the sponges are bisexual or monoecious or hermaphrodite. These terminologies are very, very important. while defining the sponges, and they are asked interchangeably. When you are asked about bisexual, it means the same. It is asking about the monoecious and hermaphrodite as well. But there are in organized gonads. Generally, archaeocytes act as gonocytes and form haploid gametes. So there is a haplontic reproduction. And there is generally internal and cross fertilization. Many sponges are protogynesis. Example, hypha. Development is always indirect, includes free swimming paracumula, solid lava in leucosolenia or amphiblastula, hollow larva, half flagellated. And what is amphiblastula larva? This is very important, which is a hollow larva, half flagellated and half non-flagellated. And in where it is found in Scypha for dispersal. Peculiar feature of development of sponges is inversion of blastula. This is very important. Inversion of blastula means that टर्निंग इनसाइड आउट। ये क्या करता है? एक्चुअली ये जो फिनोमिना है ना, ये पाइला में भी देखा जाता है, एप्पल स्नेल में भी देखा जाता है और इसमें भी देखा जाता है। ठीक है, ब्लास्टुला जब वो लावा फॉर्म हो रहा होता है ना, तो वो क्या करता है, अपने आप को ना ऐसे मोड़ लेता है इनसाइड आउट, मतलब जो अंदर वाली जो ऑर्गेन होगी वो बाहर को आ जाएगा, जो बाहर वाला होगा सरफेस वो अंदर को चला जाएगा। And flagellated layer form inner coenoderm and non-flagellated form outer pinacoderm. So the inner layer is called coenoderm, which is the flagellated one, inner layer to maintain the water current, and the outer one is non-flagellated layer, which form the outer pinacoderm. Now let's move to the unique features of phylum Porifera. First is cellular level of organization, presence of sphenoidal cycloid or leuconoid canal system. Fourth is presence of collar cells or conocytes. Fifth is endoskeleton is formed of spicules or spongin fibers or both. Sixth is absence of mouth. They don't have a proper mouth. And digestive cavity. Digestion is intracellular. And endoskeleton is formed of spicules or spongin fibers or both. Absence of mouth. Okay, this one is done. Presence of central spongiosole or paragastric cavity. Inversion, I told earlier, occurs during development, during the larval stage. Gastrulation is formed by invagiation. Okay. Fate of germ cells is opposite to that of higher animals. For fate of germ cells opposite क्यों है? क्यों? क्योंकि वो नॉन फ्लैजिलेटेड सेल फॉर्म करता है, तो वो दो तरह से करेगा, ये बाहर वाली पिनाकोडर्म हो जाएगी और जो अंदर वाली होगी वो कोएनोडर्म हो जाएगी। अंदर वाली फ्लैजिलेटेड होगी और बाहर वाली नॉन फ्लैजिलेटेड होगी। ओके, नाउ let's move to the classification. So they have three classes: calcispongiate, hyalospongiate, and dermiospongiate on the basis of habitat, size, symmetry, canal system, and nature of the endoskeleton. They are classified. So this is a very easy thing. All are marine. Let's discuss the common features. All are marine. Size. The calcispongia have small size. Hylospongiate, also called hexaceae, but the easy name is hylospongiate, so you can remember only the hylospongiate one. But if it asked about the hexaceae, so you can remember this one as well. Now next is small size. These are small size, these are moderately, and these are large size. Remember from the name demo. Demo means large size. Okay. Hilo means not very high size, so moderately size. Now all these two are radially symmetrical and this is asymmetrical. Flat, rounded, irregular, and asymmetrical. Example are Ascon type of canal system. Example Leucoselenia, and Syscon type canal system. Example Slifa, or Leucon type. Example Lucella. I have discussed the three canal system. So these are these three main canal system are there. In hylospongiate, Euphylus leucon type, a type of canal system which is out of your syllabus, but this is a kind of more advanced from this is found. Leucon type, Euphylus type, and Aphodial type, diploid type. You can just remember the name because these canal system are not in your syllabus. Okay. The next is endoskeleton, formed of only calcareous spicules, which may be monoaxon, triradiate, and tetraradiate. So I have mentioned that the spicules can be of three types, more than three types, of course, but mainly the three types are mentioned: monoaxon, having a single axon; triradiate, radiating in 3 directions, tetraradiate radiating in 4 directions. All the spicules are megasclerite. Now the next is 6-rayed hexa. Here it is hexacerite siliceous spicule. Spicule are both megascleres and microscleres. Okay. Here 8 layers of siliceous spicule or spongin fiber are present. So here maybe 1, 3, 4, here maybe 6, and here maybe 8. Examples are Leucosolenia, Scypha, and Grantia. Hylospongy example are Euplectella, Hylonema, and the last one is Silona, Chelina, Spongia, and Euspongia. Now, let's move to the canal system. Here, a good diagram of canal system. You can draw it in your notebooks. Okay, this is very important, canal system. Definition is canal system, also called aquiferous system, of interconnected canals of different types present inside the spongy body. Components of cycloid canal system. Scypha has cycloid canal system and is formed of following parts. First one is dermal ostia. What is dermal ostia? I've mentioned it, earlier, also called as inhalent or incurrent pores. A group of 3 or 4 pores in each group, small circular apertures. These are small circular apertures are present in the grooves lying between the polygonal elevations. Polygonal means many, more than one elevations. Elevations mean कि एक लेवल ऊपर, ठीक है, ऑफ द बॉडी सरफेस, एक बॉडी सरफेस से थोड़ा सा एक लेवल ऊपर पे बहुत सारे पॉलीगनल एलिवेशंस होते हैं. These are present in thin pore membrane which encloses the incurrent canal externally. The dermal ostia are surrounded by contractile cells called myocytes. Whenever the term myo comes, it means muscles. Muscles का काम क्या होता है? Contraction and relaxation, which regulate their opening and closing. These act as mouth for ongoing water currents. See, this is radial canal, this is myocytes, this is pore membrane, dermal ostia, and radial canal again. And this is one is very important. This is the Scypha. Diagram of Scypha. As you can see that oscular fringe is there. You can draw this. This is a very important diagram from exam point of view. This is the osculum. This is the dermal ostia. Dermal because on the body surface. Incurrent canals. Okay. Prosophiles. Prosophiles are very important. Prosophiles are these are surface in between the incurrent canals. Okay. Prosophiles are very important. They can be asked what are prosophile. Proso means near, phile means gate. Okay. There are short, narrow canals which are interconnected by incurrent canals with radial canals. And these are intercellular and not intracellular as in Leucosolenia. Okay. Now, this is the spongiosole. These are apophile. This is a mesenchyme, pinacoderm, coenoderm, spicules, I've told earlier, and this is a substratum. Substratum वो सरफेस होता है जहाँ पे ये रहते हैं, खड़े रहते हैं. ये प्लांट लाइक कैरेक्टर शो करते हैं कि एक जगह ही रहना है. इनकरेंट कैनाल्स क्या होते हैं? इनकरेंट कैनाल्स होते हैं इनहेलेंट या एफरेंट कैनाल्स आर नैरो एंड एक्वेरिश कैनाल्स, स्क्वेरिश, दे आर स्क्वायर शेप. They are formed by invaginations of surface. Of which surface? Ectopinacoderm and the end binding inner surface just out spongiosole. Last but not the least, radial canals. These are very important class, and this is a radial canal. Okay, so this is a purview of radial canal. This is a prosophiles. Remember, don't forget to mention this. These are prosophiles. These are incurrent canals. These are radial canals, exocurrent canals, gastric, ostia, and apophile. Apophile is the middle one. And this is the paragastric cavity. These are the outer one. This is the ostia. Okay, this is a TS, Scypha, transverse section. Radial canal are also called flagellated canals and are wider and octagonal in shape. Octagon, because they are 8. 1, 2, 3, 4, 5, 6, 7, 8. Okay, you can count. And widely octagonal, which is extragonally closed just to inner of the polygonal self elevation by internally opens in the excurrent canal by apophile. Opening is called apophile. It is lined by a layer of flagellated cells or collar cells or coenocytes. These are formed by evacuation of wall of spongiosole of asconite system. The incurrent and the radial canal lie parallel and alternate each to both vertically and radially. The arrangement of these canals is such that each radial canal is surrounded by 4 incurrent canals and vice versa. Now, Let's move to the next topic. Apophiles. Apo means away, file means gate. So the gate which take away the things out called apophiles. Also called internal ostia are opening of radial canal into the current canals. Each apophile is a large circular aperture in the center of thin portion called diaphragm. Again, now diaphragm. What is diaphragm? Here it is the diaphragm. Lying between the radial canal and excurrent canal. Its size can be regulated by it. So size is not very, you cannot say that only this will exhibit this kind of size. Okay, its size can vary. Also called myocytes. These are muscle contraction cells surrounded by apophile and regulate the opening and closing. Apophile regulate the opening and closing of canal. Now, I think this is the last one. Excurrent canals are also called exhalent or efferent canals are small wide chambers. This is a very important diagram. Okay, very, very important. This explains the whole histology. Histology, what do you mean by histology? Histology is the study of body surface, the histocytes, muscle cells, okay, body surface. The small wide chambers which radial canals open, these are lined by pinacocytes and internally open in spongiosole by wide aperture called gastral ostium. Now let's move to the next spongiosole. Sponge means hollow, also called paragastric cavity or atrium, a narrow and tubular cavity present in the center. Spongiosole. Okay, so now the paragastric cavity or atrium or cloaca is narrow and tubular cavity present in center of each branch cycon. It is lined by flat and polygonal pinacocytes. Spongiosole of different branches are continuous at the base. Osculum. Osculum are also called the exhalent or excurrent aperture. It is a large circular aperture at the tip of each branch. It is guided by a group of monoaxon, needle or shaped spicule, which collectively form a oscular fringe. What is a oscular fringe now? Fringe means a small gap, okay, and fringe means fringe gap formed by oscules. It is also surrounded by the contractile cells called myocytes, while form a kind of sphincter, which regulates its opening and closing. Now, the physiology of canal. This is a very important thing. Okay, the physiology of canal. I have made a short trick for this. So you can remember by this also. I will explain it. Physiological cells or radial cells undergo constant uncoordinated beating and maintain a water current which follows the following path. So the outside water enters in dormal ostia, which enters the incurrent canal. Then the prosophiles regulated through the radial canals. Then epiphiles regulate through the excurrent canals. Then now the thing reaches the spongiosole through gastral ostia and finally outside the osculum. Starting from in dermal ostia to ending to the osculum. Outwater, DO means dermal ostia, IC means IC, RC. Incurrent canal, radial canal, excurrent canal, spongiosole, and outwater. This is the osculum. This is the gastral ostia. Now, APO, apophyte, AGO, A for apophyte, and DOP for, this is the DOP, P for prosophile, DO for dermal ostia. So this is how I made the trick. Brings food for nutrition. So what are the significances? Brings food for nutrition, bring oxygen for respiration, bring sperm from one spoiler for reproduction. This is sexual reproduction. Outgoing water current also help in carrying out the undigested food for digestion and carry out nitrogenous waste through excretion. Now let's discuss the MCQs. Canal system is a peculiar feature of parazoans. Tissues are absent from the body surface of sponges. Porifera is not characterized by protoplasmic organization. Euspongia or bath sponge belongs to the class Demospongiae. Inhalent apertures for water current in sponges are called dermal ostia. Now let's discuss the true or false. Nidoblasts form lining of spongiosole of the Syscon. False. Why? Because nidoblasts are characteristic of nidaria and not porifera. Spongiosole of Syscon is lined by choanocytes. Next is amphiblastula larva is found in the development of most of the sponges. True. Apophytes lie between the radial canal and excurrent canal of the canal system of the sponges. False. Why? Because prosophiles are apertures leading from radial canal to the spongosphere. Now next is the canal system. Canal system helps in nutrition, locomotion of Syscon. False. Why? Why false? Because कैनाल सिस्टम का काम न्यूट्रिशन और लोकोमोशन नहीं होता है, मतलब ये होता है बट साइड वाइज। मेन काम क्या होता है? वाटर सर्कुलेशन और फीडिंग, नॉट लोकोमोशन। लास्ट इज sponges have a primitive type of nervous system. False. They lack primitive type. They lack a true nervous system and found in very rudimentary state. बहुत ही rudimentary उनका nervous system होता है. Now fill in the blanks. Cnidarian cells. So cnidariocytes are absent in sponges because they are found in outer layer. Internal buds of sponges are called gammules. Class of phylum Porifera characterized by presence of 6 ranges siliceous helices. किसमें थे siliceous? Hexaspicules, hexas, hexa, हाइलोस्पोंजिया भी बोल सकते हैं उसको। और जो लास्ट वाला था ना डेमोस्पोंजिया, उसमें 8 थे। ठीक है। Apertures between the incurrent canals and the radial canal of canal system are called prosophiles. Very important. Syscon has a syconoid canal system. Now, very short answer type questions. Which type of body plan is found in spongia? Cellular level of organization of cellular aggregate body plan. Okay, and what do you mean by it? You explain it yourself. Try it yourself. What are the poriferans called sponges? Why are they called sponges? Because the bodies are characterized by a porous structure with numerous pores, ostia. So what do they help in? They allow water to flow through a canal system. Now define the choanocytes. Define it yourself. What are called? They are also called collar cells. Name the different types of amoebocytes found in the body wall of spongia. What are the different types of amoebocytes found in the body wall of spongia? Main types of pores are ostia and osculum. Ostia are incurrent pores, osculum is excurrent pore. Ostia allow water enter, osculum allow water to exit. And the answer to the next question is here you have to explain the canal system. I have explained in short, but in exam you have to properly explain it. So the canal system is a network of canals and chambers within the sponge's body that facilitate water flow. It is also called the aquifer system because it is responsible for water circulation through sponge, bringing food and water and removing waste. Answer to the seventh question, the types of the canal system. So the first one I have discussed earlier, asconoid type line the entire spongosphere, syconoid type line the radial canal, and eluconoid type line the flagellated chamber. I have explained briefly. You have to give the example of each. Okay. Now the answer to the next question, skeletal elements. So the skeletal elements are spicules, where they are calcareous or silicates. Now spongin fibers, example Syscon. They are also calcareous spicules and euspongin, the spongin fibers. This is the example and this is the kind of fibers. Answer to the next question of the ninth question is sponges. They are ciliar feeders because the choanocyte possesses the flagella which create the water current, drawing in water current and containing the food particles then filtered and ingested. Answer to the last question for me because the rest of the question you should try yourself. Okay, I have explained the entire chapter, so you should try yourself. Answer the 10th question, what are gametes? So, asexual reproductive structures. They are the internal buds formed by the freshwater sponges and containing archaeocytes. With this, I end the lecture. Thank you, and I will meet you in the next lecture.